Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
An artificial intelligence algorithm can determine non-invasively, with about 70% accuracy, if an in vitro fertilized embryo has a normal or abnormal number of chromosomes, according to a new study from researchers at Weill Cornell Medicine. Having an abnormal number of chromosomes, a condition called aneuploidy, is a major reason embryos derived from in vitro fertilization, IVF, fail to implant or result in a healthy pregnancy. One of the current methods for detecting aneuploidy involves the biopsy-like sampling and genetic testing of cells from an embryo, an approach that adds cost to the IVF process and is invasive to the embryo. The new algorithm, STORK-A, described in a paper published December 19 in Lancet Digital Health, can help predict aneuploidy without the disadvantages of biopsy. It operates by analyzing microscope images of the embryo and incorporates information about maternal age and the IVF clinic's scoring of the embryo's appearance. Our hope is that we'll ultimately be able to predict aneuploidy in a completely non-invasive way, using artificial intelligence and computer vision techniques, said study senior author Dr. Ayman Hajirasoliha, Associate Professor of Computational Genomics and of Physiology and Biophysics at Weill. The study's first author is Josue Barnes, a doctoral student in the Weill Cornell Graduate School of Medical Sciences who studies in the Hajirasoliha laboratory. Dr. Nikica Zaninovic, Associate Professor of Embryology in Clinical Obstetrics and Gynecology and Director of the Embryology Laboratory at the Ronald O. Perelman and Claudia Cohen Center for Reproductive Medicine at Weill Cornell Medicine and New York Presbyterian, Weill Cornell Medical Center led the. According to the U.S. Centers for Disease Control and Prevention, there were more than 300,000 IVF cycles performed in the United States in 2020, resulting in about 80,000 live births. IVF experts are always looking for ways to boost that success rate, to achieve more successful pregnancies with fewer embryo transfers, which means developing better methods for identifying viable embryos. Fertility clinic staff currently use microscopy to assess embryos for large-scale abnormalities that correlate with poor viability. To obtain information about the chromosomes, clinic staff may also use a biopsy method called pre-implantation genetic testing for aneuploidy, PGTA, predominantly in women over the age of 37. To develop a computational approach to embryo assessment that capitalized on the embryology laboratory's pioneering use of time-lapse photography, investigators from the Center for Reproductive Medicine teamed up with colleagues in the Englander Institute. In a 2019 study, the teams developed an artificial intelligence AI algorithm, STORK, that could assess embryo quality about as well as IVF clinic staff. For the new study, they developed STORK-A as a potential replacement for PGTA, or as a more selective way of deciding which embryos should have PGTA testing. The new STORK-A algorithm uses microscope images of embryos taken at 5 days past fertilization, clinic staff's scoring of embryo quality, maternal age, and other information that is normally gathered as part of the IVF process. Because it uses AI, the algorithm automatically learns to correlate certain features of the data, often too subtle for the human eye, with the chance of aneuploidy. The team trained STORK-A on a dataset of 10,378 blastocysts for which ploidy status was already known. From its performance, they assessed the algorithm's accuracy in predicting aneuploid versus normal chromosome, euploid, embryos at nearly 70%, 69.3%. In predicting aneuploidy involving more than one chromosome, complex aneuploidy versus euploidy, STORK-A was 77.6% accurate. They later tested the algorithm on independent datasets, including one from an IVF clinic in Spain, and found comparable accuracy results, demonstrating the generalizability of STORK-A. The study provides a proof of concept for an approach that is currently experimental. Standardizing the use of STORK-A in clinics would require clinical trials comparing it to PGTA, and Food and Drug Administration approval, all years in the future. But the new algorithm represents progress on the way to making IVF embryo selection less risky, less subjective, less costly and more accurate. This is another great example of how AI can potentially transform medicine. The algorithm turns tens of thousands of embryo images into AI models that may ultimately be used to help improve IVF efficacy and further democratize access by reducing costs, said co-author Dr. Olivier Alemento, director of the Englander Institute for Precision Medicine and a professor of physio. We believe that ultimately by using this technology we can reduce the number of embryos to be biopsied, reduce the costs, and provide a very good tool for consultation with the patient when they need to make decision whether to do PGTA or not, said Dr. Zaninovich. The team now plans to build on this success with algorithms trained on videos of embryo development. By using video classification, we can leverage both temporal and spatial information about the embryo's development, and hopefully that will allow the detection of trends in development that distinguish aneuploidy from euploidy with even higher accuracy, Barnes said. This technology is being optimized with the hope that at some point its accuracy will be close to genetic testing, which is the gold standard and is more than 90% accurate, said co-author Dr. Zev Rosenwachs, director and physician-in-chief of the Ronald O. Perelman and Claudia Cohen Center. But we realize that this goal is aspirational. Hasway Barnes, Matthew Brendel, Vian R. Gao, Suraj Rajendran, Junbum Kim, Chianzi Lee, Jonas E. Malmsten, Jose T. Sierra, Pantelis Zissimopoulos, Alexandros Sagaras, Pega Khosravi, Marcos Mesaguer, Qianshang Zhan, Zev Rosenwachs, Olivier Alemento, Nikicha. A non-invasive artificial intelligence approach for the prediction of human blastocyst ploidy, a retrospective model development and validation study. The Lancet Digital Health, 2023. 5. 1, E28 doi. 10. 1016, S2589, 750022, 00213, 8 Weill Cornell Medicine. Harnessing Artificial Intelligence Technology for IVF Embryo Selection. Science Daily. Science Daily, 20 December 2022. ift.tt/p6qmes2. Weill Cornell Medicine. 2022, December 20. Harnessing Artificial Intelligence Technology for IVF Embryo Selection. Science Daily. Retrieved December 20, 2022 from ift.tt/p6qmes2 Weill Cornell Medicine. Harnessing Artificial Intelligence Technology for IVF Embryo Selection. Science Daily. https://ift.tt/p6qmes2. Access December 20, 2022. Thanks for watching. Please subscribe my channel for more videos.